said, you're going to be here June 12th. I said, we want to bring a check. I'm sure David was going to bring some. If I just mentioned getting a copy. Project person who's managing.
But I only heard that
One Hirschberg project is going to start. You gave them your credentials and they never replied. Because it was at the meeting.
You should follow.
Used again with that particular Golgi apparatus. And if you're like me and you think about the amazing diversity of glycoconjugation, the fact that a given protein can have glycolytic glycosylation depending on spatial where it's located. So Carlos went through the faculty promotion at St. Louis University School of Medicine. He came to UMass Medical Center in 1987, was there through 1998, and then came and was the founding chair at the Department of Molecular Cell Biology here at the dental school. So he's very widely published and regarded in the field of glycobiology, and I'm looking forward to your talk, Carlos.
Thank you. First, thanks for inviting me. Second, let me say, this is just the right. I don't want you to fall asleep. One of the things I like is there's a lot of cases I don't know. So, but I'm always present with the same dilemma. There's some people that really know what we do. We know why, they know why we do it. And then there are others that have never heard of the subject, have never even worried about this problem. So I'm going to give you some background. I'm going to go over material that is, I consider, classic within the field. And two are model systems to study biology. And what I'm going to tell you a lot is actually a combination of biochemistry and biology. So this is, as you see here, it's a cow that has fused vertebra that you see here, and has scoliosis, and has this, and you have to do a functional assay. And whenever we want a functional assay, what we do is we try to express the putative transporter in yeast, because I've shown you that the mammalian ones can be, and then isolate Golgi vesicles from yeast that has only cerevisiate 2 endogenous transporters, and then just throw the whole battery of radioactive nucleotide sugars to these transformed yeast vesicles and ask, do any of these other nucleotide sugars enter? It's a simple question, and what we found was a real surprise. One of the nucleotide sugars showed the same value in the mutants that were, when we transformed with mutants.
So no galactose, one galactose, 2 galactose changes, it shifts toward G0. So you have a putative loss of galactosylation. And then when the disease is treated successfully, the ratios return. My question is if that change in galactosylation is in lupus rheumatoid arthritis as a transporter.
I don't think anyone has looked at it, at least as far as I know. But it certainly, until someone looks at it, I don't know the answer. We can come back to that because some years ago in MDCK cells, we found that there's a mutant in UDP galactose transport in MDCK cells. Is there one pore that gets them all out? And honestly, no one has any idea what's going on. So then we wanted to look at what about glycosylation in these particular mutants. And what we did is we silenced this transporter in the procyclic form. And the main protein on the surface of the procyclic form is, of course, called procyclin. And these, before you look at this, actually, this is much better. Whether you permeabilize or not permeabilize these T. brucei, you can see that there is significantly less expression of procyclin on the surface of the silenced cells. And then we looked in the bloodstream form. And in the bloodstream form, using lectins, I'll show you the most drastic effect.
About specificity, what's the base of specificity of this stuff?
No, we don't. And the truth is, to know that, you need to go know a three-dimensional structure. And I'll tell you, we haven't put all our soul in, but we've spent a lot of money and time by individual people trying to get enough material then to collaborate with someone.
Form like a barrel, like a channel in the middle. And then they have regulating proteins, which are on the top as a plaque of the channel. And then the regulating protein play very important role, to decide either to transport the sugar stuff to outside or, keep them inside.
For example, they work for glucanac, but not for galanac after we purify it. And that's somewhat comforting. I think the bacterial system is different, but I can't swear to it. The one I think is more similar is all the eukaryotes, yeast, whatever, and go on. Okay, well, listen, thanks so much for coming.